Welcome to Genetic Mutations with Allie. Today we will be examining the products of transcription and translation from a given DNA sequence as well as the results of different mutations. Here's our DNA. The top strand is a template strand, so it is read in the 3 to 5 prime direction. The bottom strand is the coding strand, so it is read in the 5 to 3 prime direction. The template strand is read by RNA polymerase to produce our complementary mRNA strand during transcription, which is read in the 5 to 3 prime direction and shares the same sequence of nucleotides as the coding strand of DNA, but the thymine nucleotides are replaced by uracils. Transcription is the production of mRNA from DNA, whereas translation is the production of an amino acid chain that coincides with specific codons found in the coding strand of DNA. Here I have underlined segments of the mRNA strand which correspond to the codons on the coding strand of DNA. Translation of an amino acid begins and includes the start codon, AUG, and ends at the stop codon, which can be a variety of codons. Inserted here is a universal genetic code used throughout this video. As the mRNA is read in the 5 to 3 prime direction, the amino acid chain will read N-terminus, methionine, valine, arginine, phenylalanine, lysine, C-terminus. Only 5 amino acids will be read from this mRNA strand because the codon UAA corresponds to a stop codon, which signals for translation to N, and this codon is not included in the amino acid chain. The chain's net dipole will point from the N-terminus to the C-terminus because of the positive charge at the N and the negative charge at the C. Amino acids are added at the C-terminus. Now let's look at a few different mutations. First is a missense, which occurs when a mutation is in the coding strand of DNA causes the mutated codon to encode for a different amino acid than it originally coded for. In this example, guanine is replaced by cytosine, which would be a transversion, since cytosine is a pyrimidine and guanine is a purine. So when the mRNA strand is formed, it encodes for the amino acid serine instead of arginine, changing the amino acid chain accordingly. Next is a nonsense, which occurs when a mutation in the coding strand of DNA causes an early stop in translation. In this example, an adenine is replaced by a thymine in the coding strand of the mutated DNA. So this mutation is another transversion because adenine is a purine and thymine is a pyrimidine. So when the mRNA strand is formed, it encodes for an early stop in translation, as the codon that once encoded for an amino acid lysine is now a stop codon. So the amino acid chain is shortened to 4 amino acids. In a silent mutation, the nucleotide that is replaced in the mutated coding strand does not cause a change in the amino acid sequence. In this example, a cytosine is replaced by a thymine, so this mutation is a transition, as both cytosine and thymine are pyrimidines. When the mRNA strand is formed, there is no change in the original translation, as UCC and UCU encode for the same amino acid serine. Finally, for a read-through, a mutation in the coding strand causes a stop codon to turn into one that encodes for an amino acid. In this example, a thymine is replaced by a cytosine, making this another transition mutation. As seen in the newly formed mRNA strand, the codon CAA replaces the stop codon UAA, so the amino acid formed is longer than what it originally was, and translation continues until a stop codon is reached. Here are my resources. Thanks for watching.